When we begin to examine the intersection of artificial intelligence and synthetic biology, we are essentially looking at the convergence of two powerful paradigms: computational intelligence and programmable life. Synthetic biology aims to design and construct new biological systems or redesign existing ones for useful purposes, while AI provides the tools to model, predict, and optimize these complex biological processes at a scale and speed that would otherwise be impossible. Together, they are transforming bioengineering from a largely experimental discipline into a more predictive and design-driven engineering field. At the core of synthetic biology is the idea that biological systems can be treated as programmable entities. Cells, in this view, are not just living organisms but also information processing systems governed by genetic circuits. These circuits consist of DNA sequences that regulate gene expression, effectively controlling how a cell behaves under different conditions. Designing such circuits manually is extremely complex because biological systems are nonlinear, noisy, and highly context-dependent. This is where AI becomes essential, enabling researchers to navigate vast design spaces and identify configurations that achieve desired behaviors. One of the key challenges in synthetic biology is understanding the relationship between genetic sequences and functional outcomes. DNA encodes proteins, regulatory elements, and signaling pathways, but the mapping from sequence to function is not straightforward. Machine learning models, particularly those trained on large genomic and proteomic datasets, can learn patterns that link sequence variations to functional effects. These models can predict how changes in DNA will influence gene expression, protein folding, and cellular behavior, providing a foundation for rational design. Protein engineering is a central application area where AI has already demonstrated significant impact. Proteins are the workhorses of the cell, responsible for catalyzing reactions, transmitting signals, and maintaining structure. Designing proteins with specific functions, such as binding to a target molecule or catalyzing a particular reaction, is a complex optimization problem. AI models can predict protein structures, simulate interactions, and generate novel sequences with desired properties. This accelerates the development of enzymes for industrial processes, therapeutics, and diagnostics. Genetic circuit design extends these ideas to the level of cellular behavior. Researchers aim to create cells that can perform specific tasks, such as producing a drug, detecting a disease marker, or responding to environmental stimuli. AI can assist in designing regulatory networks that control these behaviors. By modeling the dynamics of gene expression and feedback loops, AI systems can identify circuit architectures that achieve stability, robustness, and desired response patterns. This reduces the need for trial and error experimentation, which is both time-consuming and costly. Another important dimension is the integration of multi-omics data. Biological systems operate across multiple layers, including genomics, transcriptomics, proteomics, and metabolomics. Each layer provides a different perspective on cellular function, and understanding their interactions is essential for effective bioengineering. AI models can integrate these heterogeneous datasets, uncovering relationships that are difficult to detect with traditional methods. This holistic view enables more accurate predictions and better informed design decisions. In the context of biomanufacturing, AI-driven synthetic biology has the potential to revolutionize how we produce chemicals, materials, and energy. Traditional manufacturing often relies on petrochemical processes that are energy-intensive and environmentally damaging. Engineered microorganisms can be used as biological factories to produce biofuels, biodegradable plastics, and other sustainable products. AI helps optimize these systems by predicting metabolic pathways, identifying bottlenecks, and suggesting modifications to improve yield and efficiency. Personalized medicine is another area where the convergence of AI and synthetic biology is particularly impactful. The goal is to tailor treatments to the individual characteristics of each patient, including their genetic makeup, disease profile, and environmental factors. AI models can analyze patient data to identify optimal therapeutic strategies, while synthetic biology enables the creation of customized treatments. For example, engineered cells can be designed to target specific disease pathways, deliver drugs, or modulate immune responses. This approach has already shown promise in areas such as cancer therapy, where personalized cell-based treatments are being developed. Synthetic food production represents a different but equally significant application. As global demand for food increases and environmental pressures intensify, there is a growing need for sustainable alternatives to traditional agriculture. Synthetic biology can be used to produce proteins, fats, and other nutrients using engineered microorganisms. AI plays a critical role in optimizing these processes, from designing metabolic pathways to scaling production. The result is the potential to create food products that are more sustainable, customizable, and resilient to supply chain disruptions. Despite these advances, the integration of AI and synthetic biology presents significant challenges. One of the primary issues is the complexity and variability of biological systems. Unlike engineered systems in traditional domains, biological systems are influenced by a wide range of factors, including environmental conditions, stochastic processes, and evolutionary dynamics. This makes modeling and prediction inherently difficult. AI models must be robust to noise and uncertainty, and their predictions must be validated experimentally. Data quality and availability are also critical concerns. While there is a growing amount of biological data, it is often incomplete, inconsistent, or biased toward certain organisms or conditions. Training effective AI models requires high-quality, diverse datasets, as well as standardized methods for data collection and annotation. Addressing these issues is essential for improving model reliability and generalization. Another challenge is interpretability. In bioengineering applications, it is not enough to know that a particular design works. Researchers need to understand why it works. This understanding is important for refining designs, ensuring safety, and gaining regulatory approval. Developing AI models that provide interpretable insights into biological processes is therefore a key area of research. Ethical and safety considerations are particularly important in this domain. The ability to design and manipulate living systems raises questions about unintended consequences, environmental impact, and biosecurity. For example, engineered organisms could potentially escape into the environment or be misused for harmful purposes. Ensuring responsible use requires robust governance frameworks, risk assessment protocols, and international collaboration. Looking toward the future, it is likely that AI-driven synthetic biology will become increasingly automated. We can envision closed-loop systems where AI models design biological constructs, automated laboratories synthesize and test them, and the results are fed back into the models for further optimization. This iterative process could dramatically accelerate the pace of discovery and innovation, moving toward a more systematic and scalable approach to bioengineering. By 2030, it is plausible that we will see widespread deployment of AI-designed biological systems across multiple industries. In healthcare, personalized cell therapies and gene-based treatments may become more common. In energy, biofuels produced by engineered microorganisms could play a larger role in reducing dependence on fossil fuels. In food production, synthetic alternatives may complement or replace traditional sources, contributing to sustainability and food security. At the same time, the increasing power of these technologies will require careful management. Balancing innovation with safety, accessibility with control, and efficiency with ethical considerations will be critical. The collaboration between AI researchers, biologists, engineers, policymakers, and ethicists will be essential to ensure that the benefits of these technologies are realized while minimizing risks. In conclusion, the integration of AI with synthetic biology and bioengineering represents a profound shift in how we design and interact with living systems. By enabling predictive modeling, efficient design, and scalable production, AI is transforming biology into an engineering discipline with unprecedented capabilities. From personalized medicine and sustainable energy to synthetic food production, the applications are vast and impactful. While challenges remain, the trajectory suggests a future where programmable biology, guided by intelligent systems, becomes a central pillar of technological and societal development.
If you find this video exciting, please like, subscribe, and turn on notifications. See you in the next AI for Beginners educational video.